Today we're going to show you how to conduct a mutagenesis experiment.
Mutagenesis is the inducing of mutations in an organism's DNA to observe the resulting mutant phenotype. This is important for determining gene function in a biological pathway, since a mutation for a specific gene can interfere with the pathway and its products, so we can determine which genes are essential for such pathways.
For this experiment, we will induce mutations in wild-type yeast colonies through UV radiation, specifically genes involved in the adenine biosynthesis pathway. The AD1 and AD2 genes help convert an intermediate known as AIR into adenine, but mutations in these genes result in the accumulation of AIR, which turns the colonies red.
Thus, through mutagenesis, we can observe the red mutant phenotype from the white cream-colored wild type and count the number of mutant colonies to calculate the mutagenesis rate.
First, we will now obtain our wild-type yeast strain. Look at how pretty it is. This pipette tip contains over 10 million yeast cells, but because this is too many and if we plate it, the plate will become overcrowded. So what we'll have to do is a serial dilution down to 10 to the 3 cells per milliliter.
We then spread some of our diluted solution onto the plate. After the dilution, we should have roughly 1,000 cells on our plate, which contains a medium to support yeast growth. Now that the cells are on the plate, even if they're too small to see, we can expose them to high-energy UV radiation, which damages the DNA and randomly induces mutation in the cells. This is how we reliably generate mutant yeast cells.
After letting the yeast cells incubate to form colonies, some of the white HBO yeast will have acquired mutations in the AD1 or AD2 genes, causing them to become red. Unfortunately, we didn't observe any red yeast in our experiment because the mutagenesis rates are so low, but we do see a dose-dependent effect of radiation on yeast survival. We then count the total number of colonies as well as the number of red colonies if we observed any. Using these numbers, we can calculate the mutagenesis rates. After counting the number of colonies, we can take the number of red mutant colonies and divide it by the total number of colonies. Multiplying that by 100 will give us the mutagenesis rate as a percent. And that, my friends, is how you do a yeast mutagenesis.